Hello again. This second part of the meiosis practical introductory video is just to acquaint yourself with the bits and pieces of meiosis and the phases that are not illustrated in the textbooks, so that you're a little bit familiar with them before we're actually looking for them under a microscope. So let's dive in and see what we find. And I think I'll start first with a little diagram explaining the nuts and bolts of what's going on in meiosis. And we start in an ordinary cell, not in meiosis, not in mitosis, in which every diploid cell has two copies of each chromosome, and one comes from the father and one comes from the mother. So in my body, there will be 46 chromosomes. Of those, 44 of them are identical. I have chromosome 1 through to 22 from my mother, and a corresponding pair from my father. From my mother, I received an X chromosome. From my father, I received a Y. So the sex chromosomes are set apart as different, but it's really interesting in locus because they receive an X chromosome from their mother, and if they're a male, they receive nothing from their father. So instead of the locus being an X and a Y, it's an X naught, so there's nothing there. Be different if we were chickens. In chickens, if you're a male, you have two similar sex chromosomes, which are called WW. If you're a female, you have two dissimilar chromosomes, so you're WZ. So it's the reverse of mammals. I think that's pretty clearly true for all of the birds. Whereas with some of the reptiles, the snakes, you've got XY, ZW, you've got in lots of lizards and crocodiles, you've got and turtles, temperature dependent sex determination, but this is a digression. So we're just talking about autosomes, the non-sex chromosomes. One derived from the mother, one derived from the father. And they have a centromere, that's quite important. This is the part of the chromosome that attaches to microtubules to be tugged to the ends of the cell. So when it divides, they're pulled apart. Because at this point, they're just growing, and then they undergo the part of the cell cycle known as S phase, which is the synthesis of new DNA. So the maternal and the paternal chromosomes are known as homologous chromosomes, obviously, same sort from each parent. So after S phase, when all the DNA is replicated, the two chromosomes now consist of two threads linked at the centromere. And so the chromosome, and it's a bit of a, it's a bit of a sad reflection that we're not a bit more creative with our naming. It's still a chromosome, but it's a different structure from before because it's now consisting of two sister chromatids. And if we were looking at mitosis, these two chromosomes would part, they'd be tugged apart at the centromere. You've got one paternal, one maternal going down to either of the poles of the cell, and it would continue identically. So in mitosis, these would separate and you have two identical cells. In meiosis, however, these two replicated chromosomes, these homologous pairs, find each other and they synapse, so they bits of each other find them, and they slowly zip up so that they become a single entity. That is called a bivalent or bivalent. I don't care what you call it. Some people are very precious about these things. So we have a new structure, still called a chromosome, instead of it being one structure as it was before S phase, or two structures as it was after S phase. It is now four structures because the replicated chromosomes, homologous chromosomes, have found each other and paired, and that is called a bivalent or bivalent. And interestingly, a protein, a special meiotic protein, forms between the two homologs. Along the protein are little nodules, you can only see them with an electron microscope, that make and heal double-strand DNA breaks. That's a bit of an oversimplification, but it'll do for our purposes. And then the sister chromatids slowly peeled apart, but the bits that have been broken don't necessarily heal with the same bit. They heal with a bit from the other homologous chromosome, the other chromatid from a homologous chromosome. So they cause shapes to form as they separate, and these shapes equate to the new combination of alleles. So they're called meiotic figures because you can see where their crossing over points have been, where the chiasmata have formed. So this slightly untidy diagram just shows a single chiasmata forming with the lower sister chromatid of one homolog and the upper sister chromatid of the other. This is not what it looks like down the microscope. And I'm going to obscure the text here, but these points of crossover are called chiasma, chiasmata, and there may be many of them, but one is illustrated for just so that it makes life a little bit simpler. And then when the microtubules attach to the centromere and pull them apart, there is a lag in the separation where those homologs that have crossed over peel apart. Where those chiasmata have formed, they will take a bit longer to unravel. So they will drag and you get these shapes. And those shapes, that stage, is what we're looking for. So let's see what this looks like in real life rather than diagrams. So this is the reality of prophase 1. We have, at the very start, very thin threads. So if you imagine the DNA is just a soup of threads in solution, and they gradually find their nucleosomes and start wrapping around them, and the nucleosomes start shortening and fattening. This is what's happening. So we start with thin threads, very hard to tell them apart, and that is called leptotene. Lepto for thin, teen for thread. Zygotene, we have paired threads, but they haven't quite finished pairing. Actually, the technical term is they're synapsing. So during zygotene, the homologous chromosomes are synapsing until they reach pachytene, the third stage, where they are fully paired right along their length. And it's at pachytene that the synaptonemal complex, this protein, is formed that zips them together. And it literally looks like a zip. It's a tripartite ribbon that runs the entire length of the pachytene chromosome. And studied along that are these recombination nodules which facilitate the double strand breaking. I need to talk a bit more slowly. So pachytene, really important stage, and it's because this takes a long time. This can often be the commonest stage that you find in a locust lobule because it takes so long to get through. So after pachytene, the synaptonemal complex breaks down, and the diplotene stage, which is called two threads, is where the chromatids, the homologous chromosomes, are separating, except at the points where these chiasmata have formed. So you get these meiotic configurations. So diakinesis, which means moving through, is where the chromosomes are still shortening and thickening as they have throughout prophase 1, and you can still see where the chiasmata are formed, but they're a little harder to distinguish because they're so compact. And then it moves on to metaphase 1, where the tubules have attached to the centromere and they start to drag the chromosomes apart. So what might you see when you look down the microscope? Well, first of all, not this stage, that's much later, not this stage, because that's a bit later, but leptotene looks a bit like this. A spaghetti bowl of threads, very hard to distinguish, although you can see that there are threads there, but it's a bit of a quagmire, a spaghetti. In zygotine, again, not this one, zygotine, but they're paired, but you can see that not fully paired. So you can actually see some of the homologous chromosomes and the sister chromatids of the homologous chromosomes quite clearly. So that's zygotine. Interesting, I hope you see it, not relevant. And then early Pachytene, where they're still quite long, but they are distinct chromosomes. Often, especially in Orthoptera, like locusts and crickets and grasshoppers, you will see at the tips of the chromosomes there are little lumps, little nodules on there. These are just extra thickening bits of the chromosome, but that's early Pachytene. And just because I worked on it, I wanted to show you that when you disperse the DNA from the chromosomes, from the bivalent, the bivalent at this stage, you're left with a protein, the synaptonemal complex, which looks a bit like a set of tram lines. The synaptonemal complex, I don't know if you've ever come across that before. Maybe you never will again. Good trivia question. Later Pachytene sees those long chromosomes shortening and thickening. You can see those dark blobs at the end of the chromosomes quite clearly. But occasionally you can see something really interesting, and that is one of the crossovers where you can quite clearly see these strands having swapped partners. So late Pachytene, really nice, I hope you see it. Again, we want the next stage, which is diplotene. So the synaptonemal complex has gone, the homologous chromosomes are no longer paired, so they're no longer pressed together, but their shapes reveal the points at which they have crossed over. That's what we're after. We'll talk about that a bit more later. I would also point out that as this is a male chromosome, the X chromosome, so remember they only are X0, they have no pair, well they're racing ahead because they haven't had to go through all this long procedure of finding another X and pairing with it and crossing over, which would happen in the female, which is why we use males, because you want lots of cells in meiosis, but the X has shortened and fattened, and it's lost that sort of fuzzy edge because it's so compacted. So when you look at meiosis in locus, you can often pick out which is the X chromosome because it's shorter and fatter has just got on with the job a bit earlier than the rest of them. And then we move on to diakinesis. This is actually two cells together, so you could count the chromosomes. But you can see it's still mildly fuzzy, but there's still some of that meiotic figure, those shapes formed by the loss of adhesion at Pachytene with the synaptonemal complex, but still with the chiasmata keeping them together. And then they move on to metaphase 1, which when they're nice, and they're actually at their most compact here, so they're small and thick. There'll be lots of other distractions with the rest of meiosis down the microscope. So here we have two quite late anaphases. So this cell was a circular cell. It's been squashed flat on its side, and the two sets of chromosomes that are being pulled to the pole of the cell are visible. And this is anaphase 1, so it will move on to the second stage of meiosis, and all those chromosomes will separate again to form the 4 haploid products. So you might find some anaphase 2s where you've got 4 cells with chromosomes looking a bit like this. But there are lots and lots of divisions. That's not an issue. At the very least, enjoy looking at meiosis and meiotic figures. If we can find diplotenes, so much the better. But the important part is that you're getting some experience of training your eye to look at cytology, cytogenetics, the study of chromosomes. So we're back to the spaghetti here, which means that we're going to stop this part of the recording and then move on to the laboratory part.